Welcome to the African Science Media Center press briefing on why Kenya should not regulate gene-edited crops like they do transgenics or GMOs. And of course, with the increase in population and demands for food, African countries are looking for ways of boosting food production and food security. Gene editing, an approach which involves editing or altering the plant sequence, for example, to make them less susceptible to pests and diseases, is proving to be a viable option. This is because genome editing in agriculture and food is producing new improved crops and other products to boost food security, depending on the regulatory approaches taken in each country or region. Commercialization of these crops and products may or may not require approval from the respective regulatory authorities, as Dr. Tripathi will tell us. However, in most African countries, including Kenya, regulation still remains a challenge. Kenya is still debating whether to regulate the gene-edited crops as genetically modified organisms or not. Dr. Tripathi will address the press on why gene-edited crops should not be regulated as transgenic and the work her institute, which is IITA, is doing. Now, Dr. Tripathi is the director of East Africa Hub at the International Institute of Tropical Agriculture, IITA, Kenya. She heads the Eastern Africa Hub and the Institute's Biotechnology Department, where she leads biotech research at the IITA based at Biosciences for East and Central Africa, Baker Hub. Her primary research focus is on genetic improvement of banana, plantain, cassava, and yam for disease and pest resistance. Dr. Tripathi obtained her doctoral degree in plant molecular biology and worked at the University of North Carolina at Greensboro, USA, before joining the IITA. Dr. Tripathi, you have journalists from across African countries, more particularly Kenya, Uganda, and others, who are eager to hear from you why these gene-edited crops should not be regulated the way the GMOs are actually edited. Colleagues and Dr. Tripathi, you're most welcome. Dr. Tripathi, the floor is yours.
Hey everyone, and thanks Daniel for giving me this opportunity to talk about why Kenya shouldn't regulate gene-edited crop as GM or transgenic crops. So I'll just try to justify the topic given to me. Okay, so as Daniel mentioned that, major global challenges in agriculture is currently the food security. As all of you know that nearly 1 billion people going hungry in the world. By 2050, there will be an estimated 10 billion people. And in order to feed all these people, we have to double the agriculture production. But the limitation is that we have the same amount of resources. So we need to double the agriculture, but using the same amount of resources, which I mean the land, the water, the other agriculture inputs we are talking about. On top of that challenge, another challenge is also the climate change. The climate change is intensifying this challenge. We are already started seeing the negative impact of the climate change on the crop yields. You can see the pests and pathogens are becoming more aggressive, but also it impacts directly the plant yields. So, and if we talk about Africa and then the food security in Africa, actually the population of Africa is projected to be doubled by 2050. And by 2100, Africa will contribute about 82% of the total growth in the population. If you see on this graph, this blue is Africa, and you can see how vast it's growing. So what Africa need is the broad-ranging support in order to make the agriculture better. So which includes the recognition of farmer needs, high-yielding varieties, widely available and affordable planting material, and also access to the market. And with the increasing demand for food, and as I mentioned, with the limited resources, we need better and more efficient ways to produce food. And one option is like, we should use all the tools available in our toolbox, including the biotechnology and the gene editing, which is a newer tool now, I will say. So now the question arises, what is gene editing? So gene editing is a group of technologies. It's not like one or two, it's a group of technologies that gives scientists an ability to make the permanent and the heritable changes at specific sites in the genome of an organism. So that means it's like we are trying to, scientists are trying to do is like trying to make a small change in the DNA already of the organism, is not bringing anything out from the outside. So it lacks any foreign DNA. It uses the cell's own DNA repair machinery. And when I say group of technologies, so there are several technologies available like meganucleases, zinc finger nucleases, talents, and more recently, the CRISPR-Cas technology. So genome editing is a technique used to precisely and efficiently make specific changes to the DNA of a cell or an organism. So what basically it does, that these enzymes, they make the double-stranded break, and then after that, that's the only thing which needs to be done. And after that, the cell does it itself. So it's like the plant machinery of the DNA repair does the repairing, and based on that, there can be small changes. It can be small addition or removal or a little bit alteration in the genome. So it can lead to the very small change or like a full gene insert into that. So what I would like to say at this point is like genome editing in the crop improvement is not something new. It has been happening since the cultivation started. But only the difference, what is happening now with the using this new genome editing tool is like it's more precise and efficient in comparison to the older techniques. So nature has been editing the genome for a very long time. And those days when the nature edits, then, farmers select, and then the mutations are more random. And then the farmers select the varieties based on of their interest. But then in 20th century, these mutations were accelerated using the chemical mutations or the radiation mutations. So, in 1985, the radiation discovery happened, and then 1927, the first time the plant mutation were reported using the radiation, and later on it was chemical. Then after that, this transgenic came in, and zinc finger nucleases, and so on, But then in 2013, the CRISPR technology was discovered, which in fact got the Nobel Prize last year in 2020. And then even the CRISPR technology is also a fast evolving. So now, just to give a little bit idea on, what type of gene editing is, because those has some implication on the regulation, whether this technology needs to be regulated or not. So there are three types of genome editing based on the process. So once these nucleases, they cut the DNA, then the repair happens. So the repair can happen in two different ways. One is that there is, in that case, it can be like a just very small, at the target, very small changes. And this is like leads to the gene knockout or the silencing of the gene, but then there is no foreign gene integrated there. And we call it SDN1. SDN1 means the site-directed nucleases, which are these nucleases or the enzyme. And then this is a type 1. So it's called SDN1. The second is like the second way of repair is when there is a donor template available and is a homologous recombination. And in that case, it can be either SDN2 or SDN3. In SDN2 is only very small correction is done. And then that's the mutation, but then you know what correction you want to make. So, so that's the SDN2 type. And then the SDN3 type is when there is a full gene insertion is there. So it allows the insertion of the gene. And then the gene insertion can be actually from the same plant species, so which we can call cisgenic, or it can be from the other plant species as well. But then this technique can also be used for the allele replacement. So basically the SDN1 and the SDN2 types are very similar. What can also happen through the old type old mutation technologies like chemical or radiation technology, or the spontaneous natural mutation, because those are the small changes which can happen in the nature. SDN1 and SDN2 types. And SDN1 and SDN2 types are actually not regulated in many countries. So with the gene editing, there are two major biosafety concerns. One is the unwanted or unintended genetic changes in the plants, and which is maybe due to the off-target mutations. But the scientists do take care of this from the very starting of the product development, because they can minimize it during the reagent designing, when we design the CRISPR-Cas reagent at that point, or using the DNA-free genome editing, in which case you just use the protein and the guide RNA. And those protein and guide RNA complex, when you deliver to the plant cell, they just quickly create the mutation, and after that, in a short while, they just get degraded. So the reagents are there only for very short while to make some mutation, and then they are not long enough to create the off-target mutation. So that's how you can actually minimize the off-target mutation. But then the another thing is, once the gene-edited product is developed, then before there is you say that this is the final product, scientists always do the off-target analysis to make sure that there is no off-target mutation in the product, which is now going out into the field. The another concern is the transgene integration, that whether there is any foreign gene integrated into that. And then again, this is done so in two ways. If the intermediate product is transgenic or GM, then that product is like back crosses. It goes into the breeding program and it's crossed back with the wild type, so that you segregate out any transgene which is integrated, and the final product has no foreign gene. Or the scientists use the DNA-free method of delivering the CRISPR-Cas reagent, and in the end, the final product has no foreign gene integration. So scientists are really taking care of the development, and then before they give go ahead to the final product, they make sure that there is no off-target effect, and then there is no foreign gene integrated in them. There are a lot of potential benefits of genome editing, you can use it for functional genomics. That means you can do it for, it can be used for the gene discovery. You can also use it for the nutritional enhancement, like protein, increasing vitamins like provitamin A. It has been tested in bananas to do the provitamin A enhancement in bananas, which can also help in minimizing the malnutrition. Oil, starch, sugar, but also it can be used for the yield improvement, like increasing the grain weight, grain size, grain number. But there are a lot of work going on for the biotic stress resistance, enhancing the resistance against bacteria, viruses, fungi, also the abiotic stress tolerance, like drought, salt, against heat and cold, and also you can generate the herbicide tolerance. So there is a lot of potential with this technology. And then among all the different enzymes used for genome editing, CRISPR is the most, I'll say, becoming most popular for gene editing, maybe because it's more it's simple and efficient, and also it's very easy to adapt. And that's the reason maybe that the CRISPR-Cas, the two ladies, the Jennifer Doudna and Carpentier, they got the Nobel Prize in chemistry last year for this technology. So let me also give you some idea about the global overview of legislation for genome editing. So Argentina was the first country in 2015 who developed the first regulation in the world for gene edited products. And then they have like a case-by-case study. And if the product has no foreign gene integrated, they don't treat them similar to the GMOs. But the other countries, like, all the countries which are in green color, these are the countries where the genome edited crops are not regulated as GMO if foreign gene is not integrated in them. Nigeria is the first African country to issue the gene edited guidelines, which is already in place. But then there are several other countries, the countries which are in yellow. These are the countries where discussion going on for developing the guideline for the gene edited products. And Kenya is the country where the draft guideline is in the quite advanced stages for the gene edited crops. Okay, so the gene edited crops, there are a lot going on, a lot of crops which are under development at the different stages. Some are at very early research and development stages, some are at the advanced research and development stages and pre-commercialization, and currently very few at the commercial stage, which is like you can see in orange here. And then it's actually many crops are including from the cereals to the vegetable crops, the fruit crops, quite a lot going on this. And both the private sector and the public academia sector is involved. And it's very interesting to see that, public and academic institutions are involved more than the private sector. And this also shows that, this technology is quite affordable and easy to adapt. And that's why, more particularly at the early research and development stages is more going on at the public and the academic institutions. So gene editing is reality. As you can see, some of the products are either already in the market or they are quite advanced for the commercialization. Like, for example, the non-browning mushroom, which has been developed using the CRISPR-Cas, is already in the market in US and Canada. Then the high oleic soybean oil was also recently approved, and that's the first gene edited food product, the plant food product on the US market. Recently, last year, the gene edited blight resistant rice was approved by USDA as well as the Colombian regulators as non-GMO. And this year, early this year, Japan approved the first gene edited tomato rich in GABA, gamma aminobutyric acid, to fight the high blood pressure. So you can see that, it has a lot of potential for food security, nutrition security, but also for the health. And then Argentina, which is like, now many countries actually following the regulatory framework from Argentina, or actually or consulting them. And that country has also, they are doing a lot of research and many products which are very closer to the commercialization. One of them is the non-browning potato, which is now currently under the field trials in Argentina. And there they have just modified the sugars responsible for the browning process. They have just turned off some of the genes so that the potatoes doesn't turn off. Also, the higher quality of alpha is quite advanced for the release. Okay, so the question also arises, what's the difference between the GMO and gene editing? Because many people think they are the same. But they are not the same, because in the GMO, GMO use the foreign gene insertion and at the random place in the genome. And the foreign gene can be from the same plant species or from different plant species, but it can be from outside of the plant as well. Because, for example, the BT crops, the gene is coming from a bacteria. And then it's another difference is that it's at the integration or the insertion of the foreign gene is at the random position. But the gene editing is very precise, and it only targets the genes of the plant itself. It doesn't bring any foreign gene from the outside, and it's very, very precise, and it's simpler and faster of this technology. And that's why, because there is no foreign gene integrated, it's also that the gene edited products are not regulated if there is no foreign gene integrated in them, similar to GMO in many countries. Now, let me also give you some idea that what's the difference between the conventional breeding, the genetic modification, and the gene editing. So I'm giving you the example using a tomato. So this tomato, the red tomato, is sweet, but it is susceptible to disease. But then what you are interested is in the sweet, but the resistance to the disease, which is the final product here. But in order to reach this product using the conventional breeding, what you need is a parent which has the tolerant to the disease, and that parent might not be sweet. So you cross those two parents, but when you cross, immediately you will not reach to the final product, because in the breeding it will bring the disease resistant gene, but it will also bring some background things with it. And so then you have to clean up, and in order to clean up, you have to do a lot of back crossing and selection in order to get the final product. Okay, so that's why it takes a long time. But then, because it doesn't require any modern biotech lab or something, so it's a low cost, and it doesn't require any regulatory regulation. But then, if you don't have this parent with this resistance to this disease, then you can't achieve this. So if the trait is not available in the gene pool, it's not possible to do this. Then, in the genetic modification, again, is the same tomato, and you want to make it sweet, but you keep the sweetness, but then you also build the disease resistance. Then you can just bring the foreign gene from other plant species or from bacteria or wherever, and then you clone that gene and then you insert in it, and then your final product will be ready. But then this will be, in comparison to the conventional breeding, it will be shorter. It will be, I will say, medium time, but it will have the high cost, because you need a laboratory and the greenhouse facility, and which is like a biosafety level 2. And also the biosafety regulation is required, because you need to prove that the product is safe before you can put it on the market. Then in the gene editing, you can actually, the same tomato, which has the sweet but is susceptible to disease, you can actually switch off these susceptibility genes in the same tomato to make it resistant to the diseases. And so that means, you have to just tweak little bit the DNA of this tomato to make it sweet. You don't bring anything from the outside to these tomatoes. So the time is shorter here in comparison to the other two technologies. It's a low medium cost, because it still requires the lab, but it doesn't require much of the regulatory science and not that much of the risk management and all those things. So that's why the cost is low. And then there is no regulatory issues because, you know, it's the same tomato. You are not bringing anything from the outside, no foreign gene there. And only there are some small biosafety concern which scientists normally take care before the product is released, like, you know, to make sure that there is no off-target mutation and there is no foreign gene integration there. But then the regulation depends upon the country policy. So it is very different in different countries, how to treat them. So this is more like a case-by-case studies. So now I will give you example of this so that you understand why the gene editing is required and how we do it. So I will give you the example of banana because that's one of the crop we are focusing on. And why we are focusing on banana is because banana is one of the major staple food crops grown in over 136 countries. Actually, majority of the countries, banana is a fruit. But when it comes to the Africa, actually banana is a staple food. And over, like, you know, global production is 153 million tons, but 1/3 of that banana comes from East Africa. And so East Africa is not only the largest banana producing, but also the largest banana consuming region, particularly when we talk about Uganda and Rwanda. But when you come to the yield, you see, so this is like a matuke varieties, which are the cooking type of bananas. The purple is the bar, which is like what the yield you are getting. So this is in the range of 5 to 30 tons hectare per year. But what the potential this crop is about 70 tons per hectare per year. So this is a huge yield gap here. Right, the same thing for the plantain. Plantain is a type of banana which is used like for roasting and is quite is a staple food in West Africa. So the current yield is about 7.8 tons per hectare per year, but the potential is the 35.5 tons per hectare per year. And some of the countries like India, this yield gap is shorter in comparison to the yield gap in Africa. So you might be wondering why there is this huge yield gap in banana. Because of this. So banana has lot of diseases and pests. So they have fungal diseases like black sigatoka, fusarium wilt, they have the bacterial wilt, the viral diseases like banana bunchy top virus, banana streak virus, and they also have the pest like nematodes and weevils. So having these many diseases and pests is not the problem. The problem is that majority of times, all these diseases and pests are present together. So they are coexisting on the same, not only in the same country, but on the same field. Many of the time you go to the banana field and you find majority of these pathogens and the pests together. So what in the graphs you can see that, you know, the different color dots means the different pest pathogen, and you can see majority of the countries have all the dots and of all the different color dots present there. So that's why the genetic improvement of banana holds great prospects for improving the food security. And there is a critical need to develop the improved banana varieties with broad spectrum resistance to various diseases and pests. And in order to do that, we have to use all different tools available in our toolbox. And so at IITA, in our banana improvement program, we use all the tools. We straightway don't jump on the gene editing, because gene editing is not a silver bullet which will solve all the problem. Is just one of the tool, which is a powerful tool, which is also giving you ability to make the improvement. So we start from the selection of the resistant varieties. We also do the conventional breeding if that's the feasible approach. And then if the conventional breeding is not, then we complement the conventional breeding using either the genetic modification or the gene editing. So we are actually working on several traits on using genetic modification, but also several traits using the gene editing. And I'll give you the example of two of these ones, the banana streak virus and the banana xanthomonas wilt. So what I was explaining there, that's what I can show you with the picture here in this slide. So basically the conventional breeding, IIT has very strong conventional breeding program. And for conventional breeding to have an improved variety, it takes 10 to 15 years. So it's a long time. As I said before, is a low cost. There is no regulatory issues required there. But then, for example, for the banana Xanthomonas wilt, there was no resistance in any of the cultivated varieties or any of these improved parents. So that means we cannot go using the conventional breeding. So then there is a genetic modification. That means, you know, if you want the disease-resistant banana, you bring the resistant genes from other plants, like you can bring it from green pepper, which we were doing that from long time, and then you insert it in banana and you do it. So this is a high cost, and it requires the biosafety regulation. But now for the disease resistance, we have a lot of information on the susceptibility genes from the wild relatives of bananas, also on the resistance gene. And then, you know, we can also transfer some of this information to our susceptible variety to make it using the genome editing, and which will be comparatively much shorter in comparison to the conventional breeding, and low to medium cost, and doesn't have much of the regulatory issues. Okay, so banana Xanthomonas wilt, this is like a very big problem, in particularly in the East and Central Africa, because it can lead to the production losses from 40 to 80% in the Great Lakes region of Eastern Central Africa, which is the biggest region for the banana production. And once the disease is there in the field, that's how the plants will look like. You know, it's like a complete wilting, and is almost the absolute loss of the banana yield, because the disease is spread very fast. And because like once the disease is in the field, within few months time, the whole field gets wiped off because of this disease. And as I was telling before, all the varieties of bananas, they are susceptible to this disease. Is only that there is a wild type of banana, Musa balbisiana, which is found to be resistant. And then there are some other wild type bananas like Musa acuminata species, which has some tolerance. But the Musa balbisiana wild type of banana is normally not used in the breeding program. So even though it has the complete resistance to the disease, it cannot go into the conventional breeding program for making the resistant varieties. And maybe I will tell you later why breeders don't use the Musa balbisiana into the breeding program. This control of BXW disease is really required, because the overall economic losses were estimated between 2 to 8 billion over a decade. And once this pathogen attacks the bananas, there is an absolute loss, and all the varieties are susceptible. So what you see in the picture down there, that's the only remedy, that, you know, once the bananas are diseased, you have to uproot the bananas and leave the field fallow for a few months before the new bananas can be planted there. Okay, so what we are trying to do here is using gene editing, is that the Musa balbisiana, which is a wild type banana, but which is also the progenitor, like one of the parent, because the banana has come from the two different parents. One is the Musa balbisiana, which has the BB genome, and the Musa acuminata, which has the AA genome. And then these two parents have crossed, and then the cultivated varieties are triploid. So they are either AAA, or they have the AAB genomes, or ABB genomes. So now what in our lab, actually, we have studied this wild type banana, which has resistant to the disease very well at the molecular level. So we have all the information available, that what are the genes which are responsible for the disease development. We call them susceptibility genes, but we also have the information about like what are the defense genes available, which are switched on in the wild type bananas, but these genes are switched off in the susceptible varieties. So what we are trying to do is we are either switching on these genes in the susceptible varieties based on the information or of the wild type, or we are switching off by knocking down of the susceptibility genes. And then these susceptibility genes we have identified based on the information from the Musa balbisiana, which is a wild type banana. And so that's the information we are utilizing for just tweaking the genome of our variety of our interest, which is susceptible to the BXW disease, and we are making them resistant to the BXW disease. So I'll just show with one gene. We are testing several of them, but one of them is the DMR6, the Downy mildew resistant 6, which is a susceptibility gene. And this gene is actually activated when there is an infection to the plant. So what we have done is we have knocked down this gene to make it switch off. And then we have created these plants, and then we have tested these mutants in the glass house after doing all the molecular characterization by sequencing to see the target mutations. And then we have tested them in the glass house. And as you can see, the bottom picture, this is our wild type bananas, non-edited. And they got the symptom when we have challenged them with the bacteria. But then the first picture is of the gene edited mutant bananas. And once we have challenged them with the bacteria, they didn't develop any symptoms, because these are the plants where the DMR6 gene is switched off. And so we have also tested them for to see if switching off this gene has any effect on the plant growth, and we have noticed no detrimental effect of this switching of this gene into the bananas, but we still need to test them under the field trials, because currently this is just in the research under the glass house. Okay, the second example I will give is about the banana streak virus. So banana streak virus is a Bedna virus, double-stranded virus, which integrates into the banana genome. So what happens is that when this virus integrates into the banana genome, it integrates in like a multiple copies. And then when it remains as integrated, the plant doesn't show any symptom. But under the stress condition, when the plant is stressed, the stress can be the environmental stress, like increase in the temperature or drought, or even because banana is a vegetatively propagated crop, it's multiplied in the tissue culture. So tissue culture is a stress as well. And then when the breeders cross these bananas, that hybridization is also plant treats it as a stress. And under that stress condition, this virus get activated into this circular form, and then they start developing the symptoms, as you see on this leaf, you know, the yellow streaks, and that's how these symptoms are. And so this is only the problem in the bananas which has the B genome in them, because this virus is integrated only into the B genome. And that is the reason that, you know, the breeders cannot use this diploid Musa balbisiana in breeding program, because it has the BB genome. And because of this virus, actually the plantain hybrids, you know, dissemination of plantain hybrid is very difficult, because it is a germplasm dissemination issue. But also the challenges is the breeding, you know, making the plantain hybrid. So that's why, you know, we worked on for making the this integrated virus inactive, so that they cannot turn into this episomal form. So what we did here is, like, you know, what you see, this one, we shredded the banana, not banana, the viral banana streak virus sequences here. So we created only small mutations in the virus genome. We didn't touch anything on the banana genome. So we created 3 different mutations in the. So there are 3 open reading frames of banana streak virus, and we just created three mutations in them. One is in the open reading frame 1, and the open reading frame 2, and the open reading frame 3. And then, so we have delivered the CRISPR-Cas reagents into the banana cells and regenerated the complete plantlets. And then these plantlets, first we did the molecular characterization, like PCR sequencing to identify the target mutations. We also did the sequencing to check for any off-target mutation, and we didn't find any off-target mutations in them. And after that, we tested these mutants into the glass house, where we have actually mimicked the drought. And so, you know, these are our potted plants, we didn't water them. And so this is the first is our control. You can see very severe streak virus symptoms on those ones. And this this is our mutant. You see no symptoms, and even here there are no symptom. But then these 2 plants also showed the mild symptoms. So what we noticed here, that all the mutants which has the mutation in the 3 sites, the target sites which we made, then those ones didn't show any symptoms of this virus. So we have finally made this virus inactive in this plant, and the plants were like resistant to this virus. And so currently what we are doing is we are improving the parents. So we are improving this 4x hybrid as well as this 2x improved, so that when we have the 2 improved parents, we will cross them to make this 3x hybrid. And this hybrid will have the banana streak virus inactivated, but will have no foreign gene integrated in them. So our final product will be non-GM with no foreign gene integration, but our trait, which is the inactivated banana streak virus, will be present in that one. So finally, in conclusion, what I would like to say is that there is no single silver bullet to fix the challenges in agriculture. It requires all countries to be open to innovation and new tools and technologies. Gene editing is a powerful tool for crop improvement. And scientists normally use the gene editing technology, targeting the plant's natural DNA to make the precise changes without bringing any foreign DNA into it. And that could also otherwise happen through the traditional methods, like spontaneous mutations or chemical mutagenesis, or even the other way of mutagenesis. But in those cases, those will not be as precise as what we can do it in gene editing. And also they take much longer in comparison to the gene editing. The process of gene editing is very precise, and because there is also no foreign DNA there, so it's quite safe. As I said, there are no trace of any foreign DNA integration in the final gene edited product. And that's why Kenya shouldn't regulate gene edited crops which has no foreign gene integration similar to GMOs. In the end, I would like to acknowledge my team at IITA who is working on it, and also our partner in University of California, Davis, and the financial support from USAID and the CGIAR research program. Thank you very much.